Hi everyone! We did our project on using microscopy for detection of signals in neurological diseases and understanding the human brain. So I'm Emily Dew.
I'm Naya.
I'm Dryly. So the brain is mostly a mystery to scientists, despite the improvements in the technology that is available in the current day, and the great number of people who work in neuroscience. As a result, we find ourselves ill-prepared to treat many neurological diseases, many of which have unknown causes and detrimental effects. The brain carries out functions through the signaling, so we wish to understand how different types of signaling may be inhibited or altered in diseases. Developing a full comprehensive picture of the human brain and its activities could potentially help all of those who suffer from such neurological diseases.
So we wish to see how current methods of nanoscale microscopy can address neurological diseases such as schizophrenia. So we thought this was a relevant topic because nearly 1 billion people in the world suffer from neurological diseases, and as the world has a population of 7 billion, that's approximately 1 in 7 people. So in a social context, accurate and in-depth information from imaging can help us develop better medicines and possibly lead to cures for these diseases. So as there's a large amount of information that's unknown about what these diseases are caused by and how to possibly address them, there's a lot of fear surrounding these diseases, and additionally there's sometimes social stigma around them, especially around diseases such as schizophrenia or psychosis. And a better understanding of the brain can lead to better methods of addressing these in the future. So in the past, researchers have studied the activity of various neurotransmitters such as glutamine and dopamine. They've also studied genetics because there's been research on whether there are certain diseases that are hereditary or if certain genes make you more likely to develop a certain disease. People have also studied neuronal structure. So what we see is that in some diseases there are types of neural network breakdown, or in general the structure of neurons themselves are distorted or damaged. Additionally, people have used mouse models to study and simulate these diseases, and they've also looked at pedigree trees, in which case there are some diseases that are hereditary, such as Huntington's, and expansion microscopy to study brain tissue, and electron microscopy to connect images of the brain.
Okay, so for our project proposal, we didn't want to hone in on a specific method. We wanted to address all the techniques since the brain cannot be understood from a single perspective. So we've used all the methods that we've learned this week and come up with a couple of techniques for each of them. So for optical and fluorescence microscopy, we can map the brain using clarity methods to get past the obstacle of opacity from the lipids in brain tissue. And we can use light sheet microscopy with fluorescent dye to study like single layers of the brain and then compile an image for that. And then we can also use expansion microscopy to study proteins and networks, and this has already been done by researchers at MIT, and they've studied that in the zebrafish. And we can also use confocal microscopy for deviations between healthy and diseased neurons. And for electron microscopy, we can use scanning electron microscopy to image slices of brain tissue and compile them into like a comprehensive model of the brain, which MIT has also done with the zebrafish. And for scanning probe microscopy, there are also a lot of applications to study the brain. And so we can use biological atomic force microscopy to study misfolded membrane proteins in neurons, since these have been linked to disease. And we can also put cells on the end of the probe and study neuron-to-neuron interaction. And we can also study the nanomembrane vesicles that we learned about yesterday to look for diseased cells in the blood-brain barrier, cerebrospinal fluid, to look for diseased cells sending out bad RNA and infecting other cells. And so for our challenges, we have two main ones, like ethics and technology. So for ethics, the brain is like a center for our personal identity and consciousness. So this gives us an obstacle as to where the line is concerning safety and privacy. And also theoretical breakthroughs can give us biological consequences. So for example, take if we have a memory retaining breakthrough, then that could mess with the balance between remembering and forgetting, and that can cause a neural overload. And also cost and accessibility. So brain enhancements can, if they are developed, then they can widen the gap of socioeconomic status. And for technology, we don't have the technology available for us to know if there are more cells than we presently know about, like neurons and neuroglial cells. And there's also the issue of lipid opacity, which has been addressed with clarity. And there's also the issue of time and expenses, since these microscopic techniques are take a lot of money and time. And so for our discussion, we wish to study signaling and identify patterns in neurological diseases and try to target causes and find cures for these causes. And so these present various questions such as what are the predicted possible irregularities that cause neurological diseases, and what correlations between symptoms and causes do we expect to see. And so these are our contributions, and these are some references.
When you said you were going to use optical microscopy in part to study the brain, you talked about using clarity methods. So, like, what precisely does that entail?
Well, so the clarity methods have been developed. It's a technology where it allows us to see past like the lipids. And so, for example, like we saw like the picture in the PowerPoint, it allows us to see through to get the things we actually want to see. Like if we want to see individual neurons or networks, we can see past just the opaque tissue.
So are you thinking about trying to take brains, for example, maybe post-mortem, clarify them, and then look at, and image that entire brain or important sections of it in order to then compare the diseased brain to an undiseased brain?
Yeah, that's primarily our goal, just looking at the discrepancies between healthy brains and diseased brains. And since there's no ethical way to, say, image or model live brains, that's really what we have to work with, other than mice and zebrafish.